reactions. Okay. Okay, if we have time, I don't know if we'll have time to do the animation today, but if not, you can go and look at the animation. No. It's molecular? Yeah. Okay. Okay, anybody not get to see their exam last time? I brought them again today. Okay. Oh. It's definitely a Monday. It's like half the class in here. It's a Monday. Make sure you check in on attendance. Get your credit for being here. Got 2 more minutes. All right, y'all about ready? So don't forget to check in on attendance. Check in on attendance. Let's see if I can. See if I can put the chat up. Okay. All right, so why did the pillow cross the road? Because he was collecting the feathers from the chicken. It was kind of sad. Charles did not get it at all. I had to explain the whole thing to him. It was sort of a disaster, but that's okay. It happens. But look, it was so difficult to have a joke every single day that if it's good or bad, whatever, we're rolling with it. All right, so the last thing we were talking about were typical Michaelis-Minton kinetics, right? Remember this? We were graphing the concentration of our substrate versus the initial velocity of our reaction and said, right, as you increase substrate concentration, the reaction goes faster and faster, but up to a point. There gets to be a point where no matter how much substrate you add, the reaction can't go any faster. And we call that Vmax. And if you find half of the Vmax, and you go and find the concentration of the substrate at that half Vmax, what is that? The Km, the Michaelis-Minton constant. Therefore, it is, guess what? Constant. It's Monday. It's totally a Monday. So what do we kind of equate this Michaelis-Minton constant to? We said it's a K value. It's an equilibrium value. But what does it really tell us about? Yeah, it tells us about affinity. Yes, yes. So it's really similar to the what? KD. It's very similar to the KD, right? It's the dissociation because we're looking at the enzyme substrate either disassociating because you're making a product or the enzyme substrate disassociating because the substrate leaves the pocket, right? But it's really a KD. So if you want a really, really efficient enzyme, do you want a high KD or a low KD? A low KD, right? We don't want it to disassociate very much, right? Okay. So now what happens if we increase the concentration of our enzyme? what happens to the velocity of the reaction? It increases, it absolutely increases. But if we take half of the Vmax, we will be at that same substrate concentration. And that is why this is a constant, because the disassociation of the substrate from the enzyme substrate complex is going to be consistent, right? Because that has to do with the enzyme itself. If you have a different enzyme, oh, then you're probably going to have a different Km, right? Okay, so.
Do you want Km to be low? Or since it's similar to Km?
Yes, you want it to be low. You absolutely want it to be low. And we're gonna look at some examples. But a lot of times this is sort of difficult to look at. It's hard to compare. So if we linearize this data, if we take the reciprocal of the data, it's the double reciprocal, and we graph it, this is what we end up with. The graph on the previous slide is the same data here. All we did was do the double reciprocal of the data. You are not going to have to do that calculation on an exam, okay? You just have to understand what happened, right? So if we graph this, what ends up happening is that you can look at where these lines intersect the x-axis, and that relates to your Km. So it's very easy to look at this graph and say, oh, both lines have the same Km, right? But what's different? They're Vmaxes. Vmaxes where they cross the y-axis, okay? So you can see as we change concentration of enzyme, because it's the same enzyme, our Km is constant, but as we increase enzyme, our Vmax changes, right? And so look here, we're going from low to high because this is the reciprocal. So it should be lower on the y-axis. So just kind of keep that in mind. All right. And then we can look at the slope. The slope is another really nice thing. You can compare, because look at this formula, right? It's a linear y is equal to mx plus b, right? It's that same format. So what is m? M is really Km over Vmax. What is x? X is 1 over your substrate concentration. So it's a very nice way to compare two conditions.
So the y-intercept is it 1 over the Vmax?
Yeah, well, for Km, it's negative 1 over Km. So it's on the other side of the origin. And then for Vmax, it's just 1 over Vmax. Okay. All right. But that doesn't always tell us everything we need to know. There's another really important characteristic of an enzyme, and that's what is its maximum catalytic activity when you have saturating levels of substrate. So that means that the enzyme is able to bind substrate as quickly as possible, right? So what's the rate limiting thing? If we can bind a substrate as much as we want, what's going to be our rate limiting condition? How quickly we can go from substrate to product, right? So that's our turnover number. So it's the number of reactants to products over time. Time, right? That's a really important characteristic. So you have to think that all enzymes have unique properties. So if you know the catalytic efficiency, do you know which enzyme is going to be the most efficient under certain conditions in the cell? Not necessarily. What else do I need to know? I need to know my Km over Vmax, right? Because what if the binding affinity is low? Even though it can work efficiently, but binding affinity is low. Well, that's not going to work so well, right? So what we really need to do is really need to know not just the turnover number, but we really need one characteristic that's going to encompass everything, and that's the catalytic efficiency. So catalytic efficiency is our combination of that turnover number, how many you can make, and how easy it is to bind, right? So tell me about Kcat. Do we want Kcat to be high or low? We want it to be high. How about Km? We want it to be what? Low. We want it to be low. So what do we want our catalytic efficiency to be, high or low? Because this equals Kcat over Km. So we want it to be what? High. This you have to be able to look at and kind of tell me which enzyme or which substrate is optimal for certain conditions. And I'm going to show you an example. Okay. So here's an example. All right, let me see if I can move some of this stuff. And as soon as I put my cursor up there, it blocks half the question. Okay, so carbonic anhydrous is an enzyme that maintains buffering capacity of the blood, right? It can use two substrates. Use the table, which would be the best choice for a substrate and why. So this is the same table that's in your textbook, table 7.4. I just pulled straight from your textbook. So you're going to look at carbonic anhydrous. Here are my two substrates. Which one is the correct choice? CO2. CO2 is the correct choice. Now, the question is why? You can't just say that the catalytic efficiency is the highest. You have to tell me why the catalytic efficiency is the highest. The specificity constant is higher. Okay. Yes, no, maybe so. Yeah. Your book does one in the animation of fumarase. So they do a very, very similar thing in the animation where it talks about which substrate is better. So it's the same analogy, exactly the same analogy. Okay, so there are different conditions that can affect enzyme activity. Lots and lots of different conditions, right? pH and temperature are going to have drastic effects on enzyme activity, right? Why might pH and temperature have huge effects on our enzymes? How about pH? Let's tackle pH first. pH will change charge of what? What do you mean specifically? How does it change charge? Change the charge of amino acid side chains. And now we're really just talking about the enzyme. So it would alter substrates, but in this question, we're just talking about enzyme. That's true, ENZYME. Okay. But so what? So it can absolutely change binding affinity. It can change so it changes the chemical nature of, so changes, I'm trying not to talk, I'm trying to write, changes chemical properties of the catalytic site. True. What else? So that's how it does it. There's another way. That's absolutely true. Absolutely. If you start changing side chains, what determines the overall three-dimensional shape of a protein? The amino acid side chains. If you start changing those amino acid side chains, guess what? You're going to change the overall structure of the protein. Can change overall 3D structure of protein. What happens if we get to extreme pHs? What happens to our proteins? They end up just denaturing and they're blobs, right? So that's an extreme case of changing the overall three-dimensional shape. It still is, but it's an extreme case. So if I get too high of a pH or too low of a pH, will any enzyme work? I beg to differ. Depends, right? Look at this. If I go down to a pH of 1.6, holy moly. This is a really low pH. I have this enzyme pepsin that's super active. Look at its relative enzyme activity. Why? This is the natural environment of this protein. Can you make a prediction on where that natural environment is? It's the stomach, right? Absolutely. So it absolutely depends. These two glucose 6-phosphatase, arginase, these are in the liver, right? So look at their pH. Their pH is definitely going to be closer to neutral or in general higher. And it really does depend on even super local areas within different organs of pH. So you can't make big general statements. You have to take into account what is the natural environment. So how does this happen? How do we have enzymes that are active at pH 1.6 and ones that are active at 9.7? So it all depends on the side chains. It completely depends on the side chains. How are the side chains determined? Like, how do we know the amino acid sequence? DNA, right? So all of these changes came about through evolution. So random point mutation changes that facilitated optimal conditions for this enzyme to work in the stomach, right? So it's all through evolution, which is insanely, awesomely cool, right? Okay, so that's pH. How about temperature? In general, what happens as we increase temperature? So temperature, in general, increased temperature, increase rate of reaction. Generally, sure. But what happens if we get too hot? They just denature. So you can also have, at extremely high temperatures, you get denaturation. You have to be very careful about that, right? Because especially think about protein purification, right? There are a lot of steps where temperature gets really, really high, especially when in that initial point when you break open those cells. A lot of times you use high shearing force, which increases the temperature quite a bit. So if you increase the temperature at your very beginning protein purification step, what's going to happen to the activity of your protein? It's going to suck because you just heat killed all of your protein in the very first step, which is so sad, right? Okay. And there are exceptions to every rule, right? So most proteins work at what general temperature range? Which is why body temperature, right? But there's one enzyme that we talked about in class that's Taq polymerase. Where do we use Taq DNA polymerase? In a PCR. It's really, really, really different than E. coli, than our DNA polymerases, in that it can be active at very, very high temperatures. Why can it be active at very, very high temperatures? Where do we find the organism that has Taq polymerase? In geothermal vents under the ocean, right? So they have to be able to work at very, very high temperatures. So in this section, right, we covered 3 really big things that affect enzyme activity. Substrate concentration, pH, well, concentration in general, whether that's substrate or enzyme concentration, right? pH and temperature, those are the kind of the big 3, right? Okay, so now we get to talk about how we regulate enzymes. And when we regulate enzymes, proteins are the prime choice for enzymes because you can modulate their activity in so many different ways, right? And you can modulate them in the positive direction to be stimulatory, and you can modulate them in the inhibitory direction so you can inhibit their activity, right? So all the different mechanisms fit into 2 categories: bioavailability and control of our catalytic efficiency. So let's look at those two and look at all the different ways that that can happen. So let's first talk about bioavailability. So what did we say were some of the things that controlled bioavailability? When we go from DNA to, like, we go from DNA to what to get to protein? DNA to RNA. Well, when you say RNA, there's kind of a lot of things that have to happen, right? You have to first make the RNA. So RNA synthesis, what do we call that inside the cell? Cell transcription. Now that you've made that RNA transcript, you have to process it. Can we have different ways to process RNA? Yeah, what do we call that? Splicing, alternative splicing. Right, so that's another way that we can change the availability, because if you have different exons inside of that RNA transcript, you're going to have a different protein, so you have different possible substrate targets, possible catalytic efficiency, right? All kinds of different things. Then what? How well we can change that RNA transcript into an actual protein. So protein synthesis. What do we call protein synthesis? Translation. Lots of chemistry people in here. All right. So these are kind of our big ones on bioavailability. But even though you make this protein, what was another way that we could control how much activity this protein, this enzyme, has on its particular substrate? We can do some post-translational modifications, absolutely, but that's going to change our catalytic efficiency, right? So what's another one that's bioavailability before we move on to catalytic efficiency? If your substrate is in a lysosome and your enzyme's in the cytosol, what's your catalytic efficiency? 0, right? Okay, so what do we call that? Localization, right? Where in the cell are you delivering your enzyme to? And does it coincide with the location of the substrate? So remember we said location, 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 right? Like real estate. Okay, so that's protein targeting. So a lot of times there's a little piece on the end of the protein that basically tells it where to go. And so that helps. What's another one we've talked about? How long that protein lasts, right? It could last for a long time, or we could degrade it really, really quickly. So rate of protein degradation is another bioavailability. So all of those things that regulate bioavailability are covered in like the last 4 chapters of the textbook that we're not going to get to in this class. Shucks. But if you take biology classes, you go over all those things, right? Those things, except for really protein degradation. Everything else is covered in your like molecular biology and genetics, right? Most of it is. Okay, so how about catalytic efficiency? What determines catalytic efficiency? Well, what did we say just a minute ago is covalent modifications. What were some examples of covalent modifications that we've talked about so far? Phosphorylation is a good example of that, or dephosphorylation, right? Another one is binding of regulatory molecules, right? One example of that is feedback inhibition, right? If you make a lot of your product, do you want your reaction to continue? Usually not, right? So like, let's say you're going through, you've eaten a meal, right? You ate a meal with lots of glucose in it, let's just say. So your body uses that glucose and it makes ATP, you know, it goes through glycolysis, goes through citrate, goes through electron transport chain. You make a ton of ATP. That means your energy charge is super high. Well, your body is designed to say, hey, I've reached my maximum, right? Now I need to prepare my body for times of stress and cold, and I'm not going to eat for a long time, right? And so I'm going to, instead of turning this into ATP, I'm going to use the rest of that glucose and I'm going to go make fats, right? And that's how it works. So if the energy charge is high, we're going to inhibit the electron transport chain, right? We're going to inhibit parts of the citrate cycle. We're going to inhibit the things that we don't need, and we're going to go and make fatty acids. So that's one way that you can regulate catalytic efficiency, is having a substrate associate with your enzyme. Then the last one is proteolytic processing. So in some, this isn't super common, but it's seen. So sometimes your protein is made and it has a piece attached to it. And with that piece attached to it, it's not catalytically active. And under certain conditions, like low pH, it undergoes a conformational change and auto-cleaves itself. Now that that regulatory piece is gone, now it can function, right? So these three at the bottom are all control catalytic efficiency. All right, so now that we know those, we're going to talk about really just these down here, catalytic efficiency, and how can we control catalytic efficiency, even though bioavailability is really cool too. So there are a lot of different ways that we can inhibit enzymes. There are reversible things that we can do, and there are irreversible things. So reversible, any non-covalent interaction of little molecules to our enzyme, right? Just like what I said, ATP. ATP associates with, I don't know, an enzyme in the citrate cycle and inhibits it, right? So you can change this inhibition just by changing the concentrations of the substances in your test tube, right? If I dilute the amount of ATP there, what happens? My reaction will pick up again, right? And so there are three classes of inhibitors within this irreversible category. They can be competitive, which means they compete for the site, for the normal substrate site. It can be uncompetitive, where it binds, but it doesn't compete. And then you can have some weird mixed things going on, which is kind of interesting. In irreversible, you're going to make usually a covalent bond, not always. Sometimes it's just really, really strong non-covalent, but most of the time it's covalent. And it typically happens with those amino acid side chains that are sticking into the catalytic site. And that basically kills the enzyme. And so it can no longer do its job, right? So sometimes they call them suicide inhibitors because they bind and they don't let go. And they go down with the enzyme, right? So look at the difference here. What happens if you have a covalent binding, right? It's a suicide inhibitor. It goes down with the ship. It goes down with the enzyme. What happens if we dilute it? If I add more water? Nothing. It's a covalent modification. It's stuck, right? So make sure you know the difference between the two and why one dilution can change the efficiency of one and not the efficiency of the other. All right, so here's an example of a reversible inhibitor. So this enzyme is succinate dehydrogenase. And succinate dehydrogenase converts succinate, right, through a dehydrogenation reaction, beautiful name, into fumarate. Now, malonate is an inhibitor of succinate dehydrogenase because it will actually compete for binding to succinate dehydrogenase. So we have two substrates that will bind to the same enzyme, right? So if malonate is there, you're going to have no reaction. So in this case, this is a product, a downstream product in a reaction cycle. And so if we have this downstream buildup of this downstream product, what do we want to do? We want to inhibit the reaction from continuing, right? So this is an example of feedback inhibition, but it's reversible, right? Because let's say you haven't eaten in a while and you don't go through glycolysis and citrate, right? You stop building up this inhibitor. Because it's non-covalent, as soon as it's gone, the enzyme activity picks up again. Okay, all right. So we're going to look at the 3 different ones and kind of talk about each one and then look at some examples. So if you have competitive inhibition, we have an enzyme. Our enzyme typically binds to some substrate to make an enzyme substrate complex, and then we have a product. So in competitive inhibition, this inhibitor is going to compete for the binding. So can you have inhibitor bound to an enzyme substrate complex? No. So this is no enzyme substrate inhibitor complex, right? No, does not exist. So how are the different ways that this could happen? You could have the inhibitor bind in the active site, just like it's shown in the picture. That's what it's shown, right? It looks almost exactly the same. But it doesn't necessarily have to. It could bind near the active site and then just prevent our substrate from attaching, right? So maybe it partially blocks the binding site, but it doesn't fit perfectly. And the other thing is the enzyme can bind far from the active site. How is that possible? And still get inhibition? Conformational changes. It induces conformational changes that propagate to that active site and therefore inhibit binding of our substrate, right? Now, uncompetitive inhibition. So when you see uncompetitive, you should automatically think, yes, if it's uncompetitive, I can get an enzyme substrate inhibitor complex, right? Because if it's not competitive, that means they can both bind to the enzyme. So what you end up having is either an empty enzyme that's going to bind its substrate. So now we have an enzyme substrate complex, but this inhibitor binds to this complex. So the inhibitor binds, now the enzyme through allosteric propagated changes, something changes in the enzymatic pocket, and now we can no longer make product. But the substrate can still bind. This is still substrate. Okay? So just because you know where the inhibitor binds does not mean that you know which one it is. You have to know, can it be bound alone or can it be bound in a complex with the substrate? That's what will tell you competitive versus uncompetitive, right? Okay, mixed inhibition gets a little more complicated. In mixed inhibition, you can have your inhibitor bind to the enzyme alone, which deactivates the enzyme, or the inhibitor can bind to the inhibitor substrate complex and make enzyme substrate inhibitor complex, and therefore it's inactive. So either way, it will be inactive, but you can have a population of enzyme that's bound to the inhibitor and a population that is bound to the substrate and the inhibitor. Yeah? Okay. I feel like this was a complicated topic for a Monday morning. All right. So in competitive inhibition, right? So usually our inhibitor is going to bind at the active site, usually. But it can bind just so that it inhibits or obstructs our substrate from binding through a steric clash, right? And add in here can be far from active site, right? Because we said that. But how can we overcome this inhibition? if it's competitive and that enzyme can only bind one thing, either it's binding the inhibitor or it's binding the substrate, but only one, whichever one you have more of is what that enzyme is going to be bound to, right? So if you have more inhibitor, the enzyme is inhibited. If you have more substrate, the enzyme is active, right? Okay. So by that definition, Vmax is unchanged. What is Vmax? The maximum velocity of the reaction, right? Completely unchanged. But what happens to our Km value? Km value changes. Km value changes. Why does the Km value change? You're binding two different things, and so basically you're removing enzyme by having an inhibitor present. You're getting rid of basically enzyme. You're like pretending you have less enzyme there than you should. So we'll change your Km value. You don't have to know all the formulas. You don't have to know the formulas. Okay, so let's look at a graph, right? So the orange line is no inhibitor. If we add in a little bit of inhibitor, we're eventually, if we continue to add substrate, eventually it'll take a lot more substrate, but eventually we're going to get to that Vmax. But because we had to add more substrate in order to get to that Vmax, what happened to our Km? Km increases because we have a lower binding affinity, basically. Basically, it changes our binding affinity. If we add even more inhibitor, it's going to take an even higher substrate concentration to eventually reach the Vmax. And so we expect to see that our Km will be even higher, right? Because you're basically eliminating enzyme molecules from participating. So if we plotted this as a double reciprocal, what we should see is that our Kms change. So here's our original, and then low and high inhibitor concentration. So our Km will change, but what happens to our Vmax? Stays the same. How much easier is it to, if you know what the values are, to look at this than this? If you know what it means. If you know what it means, this is easier. Yeah. Okay. So you can have things that that are competitive inhibitors, and we've designed an HIV inhibitor. So this is one that inhibits one of the proteolytic cleavage events that are required for HIV to replicate inside the cell. So this is an inhibitor that should be very specific to the virus and not specific to any other protease inside of our entire bodies, right? And so that's the beauty of these inhibitors, is that you can make them super insanely specific. And so they target only one enzyme. And so if you can find an enzyme that is specific in a virus or a bacteria or whatever it is, and you can inhibit that, then you're golden, then you're good, right? So, so if you look at the structure of, this is a common structure that's required for the protease inhibitor to cleave a phenylalanine and proline together. And so if you look at the structure of these inhibitors, what do you see? Very similar, very, very similar structures. Now, does the rest of the molecule matter? No, not really, right? It's just a little piece of it. Okay. All right, so uncompetitive inhibition, right? This one can bind to our enzyme substrate complex. It can't bind to the enzyme alone. It's only going to bind to the enzyme substrate complex, and it's going to alter the active site conformation. So usually this happens when you have enzymes with multiple substrates, because you don't want to get into the pocket, right? It's evolutionarily better to have an inhibitor that will bind on the outside so that it doesn't interfere with all the different things that can interact in the active site. So if you look at this, what do we notice here for our Km values? All of our Km values are different, and all of our Vmax are also different, right? So can you explain why? Because you're what?
You got less substrate binding.
You do have less substrate binding to the enzyme, but the substrate, you do have different substrates, but the inhibitor can bind to the enzyme substrate complex. So your enzyme can still bind to the substrate, but once it binds, now it can't catalyze anything, right? So it doesn't matter how much you add, you're not going to dilute out this reaction, this inhibition. You're not going to change it. If you have inhibitor present, you're going to inhibit the reaction.
So does like this substrate kind of get like stuck in the enzyme or not really?
It can. Yeah. Not always, but it can. So then if you have mixed inhibition, right? These are going to bind to sites that are far from the active site, and these bind to both the enzymes. So you have inhibitor enzyme, and you have inhibitor enzyme substrate complexes. Now, this can do all kinds of crazy things. I won't test you on mixed inhibition because it's kind of mean. So it's gonna, whenever you see it, you're always gonna see a lower Vmax, and then Km can either go up or down, right? So look at your Km values here, right? What happens to these Km values? Here's your no inhibitor. Now, as we increase inhibitor, what happens to Km? If there's a negative here, right? So it's going to go in the opposite. So it's going to increase, right? And then what happens to our Vmax as we add inhibitor? Well, it doesn't really increase. It's really the reciprocal. Yeah. But I'm not going to test you on mixed inhibition. Mixed inhibition is kind of a pain. But now I'm going to say one other thing. Not all enzymes follow Michaelis-Minton kinetics. These are like simple enzymes that follow Michaelis-Minton kinetics. There are some that require way more math in order to follow their kinetic profiles. So this is kind of like the easy ones, but we're not going to go any farther than that. All right, so those are reversible, right? So if something disassociates, it can change. You can have irreversible, right? So if you have irreversible, typically we're forming some kind of a covalent bond. It can be a really strong non-covalent, but typically it's a covalent bond. So if you form a covalent bond with residues that are supposed to participate in the active site, reactive residues, what's going to happen? Right? You remember all those enzyme kinetics where we looked at each side chain? What if you mess up one of those residues? It's going to completely inhibit the enzyme, and the enzyme is going to be inactive, right? And not only is the enzyme going to be inactive, but you've taken it out permanently, right? It's a covalent bond. You're not going to disassociate it. Okay. So when we talk about these reversible inhibitors and things that can regulate enzyme activity, a lot of times what we talk about are allosteric regulations, right? So if you have some kind of an inhibitor that's going to bind far from the active site, well, how does that work? A lot of times enzymes have a catalytic unit, and then they have a regulatory unit, right? And so the regulatory unit is just there to bind to regulatory molecules and then propagate that conformational change to the active site. So in this example, right, this is an enzyme that is involved in production of CTP. So if I'm going to try to produce CTP, what I want is for my product to inhibit the reaction. So CTP will bind to this regulatory subunit, and it'll change the active site. And so the active site becomes basically inactive, right? Well, what happens if my levels of CTP drop, right? Like I need to make nucleotides for DNA, and I basically run out of CTP. Well, that means that something else is going to be there. In this case, it's going to be ATP. So if I have lots of ATP, I have high energy. So number one, I have the energy to make DNA. And then number two, I have some substrate there in order to make the CTP, because I need ATP in order to make CTP. So I need to have ATP present and not a whole lot of CTP. And that's what makes my enzyme active. Yeah? But my CTP and ATP, are they binding at the active site? No, they're binding to the regulatory subunit. So see how the regulatory subunit is a particular conformation here when it's bound to CTP? Now we have this change in that conformation, and that conformation gets propagated into the catalytic subunit of the enzyme. Yeah, okay. All right, I'm going to show you this at the end, after we talk about the other ways that we can regulate catalytic activity. So this is what we were talking about earlier. In our metabolic pathways, we want the end product to basically inhibit the initial steps. And so a lot of times what you see is that the very end product will inhibit one of the very first steps, so that you're not wasting energy and resources in order to get halfway through a pathway and then have to stop. You want to stop it as early on as possible, right? So here's our example, ACT case. So this works by feedback inhibition, and we have two molecules that are allosteric regulators, CTP and ATP, the same ones that we talked about just a second ago, right? So if you want to promote the activity of this enzyme, right? Well, what do you need? You need to make sure that you're not making too much product. So at the end of this entire pathway, ATC case is one of the very first enzymes that's required to make CTP. So you see how they have all these lines here? This means that there's step after step after step after step, and eventually you make CTP, right? So if you have lots of CTP, it is going to feedback inhibit enzyme number one in the catalytic, pathway, in the entire pathway, and stop the procedure from going over and over and over again. So this would be an example of a negative feedback inhibitor, right? And it's allosteric because there's no covalent binding. So if you look at enzyme kinetics, right? So this is our starting substrate, aspartate. So if we have, this is our normal binding curve for our reaction. If we add CTP, that's our product, what's going to happen to the kinetics of the reaction? We go from green to red. What happens to the velocity of our reaction at the same concentration of substrate? It decreases, right? And that's what we want. We want feedback inhibition to decrease. But at the same time, in every cell, you want to have positive and negative regulators, right? Because at the same time you repress something, you want to activate something else. And so a lot of times you'll see this in, oh my gosh, every different biosynthetic pathway. So if we then add ATP, something that the reaction needs in order to proceed, then you're going to actually stimulate the activity of the enzyme. And typically, negative inhibitors tend to have a greater effect than positive inhibitors. That's kind of a standard thing that you're going to see. So one of the other ways we said that you can modify an enzyme for activity is through covalent modification, right? By adding a phosphoryl group or something like that. But phosphorylation is not the only one. You can have methylation. We talked about that, right, on DNA. Did we talk about acetylation? No, yes, maybe. Adenylation. So anything where you can add a monophosphate, right? UTP, you can take one of those off, right? I'm going to show you some. But the most common modification is phosphorylation. And remember, you have 3 residues that are preferably phosphorylated. Why? because they have alcohol, they have OH groups, right? And so those are kinases that do that. And then we have phosphatases that remove it. So if you look, this is like the prime example of phosphorylation controlling an enzyme. You have glycogen inside of your liver cells, inside of your muscle cells, right? Why do you have glycogen? What is glycogen? It's a storage form of glucose, right? And so that's really, really important for if you need to get somewhere very quickly. Like let's say you're in fight or flight, right? And a bear came in the back of the classroom. I know I always use this, right? You're gonna get a surge of epinephrine in your bloodstream. What happens because of that epinephrine? Lots of different things in your body. Your eyes dilate, your breathing rate increases, your heart rate increases, all this kind of stuff. But something else that happens is that inside of your liver, that glycogen is metabolized. So it automatically, glycogen is, it's got a single core, and then it's got all these strands of polymers of glucose that basically come out and then are branched and branched and branched and branched, and it kind of ends up looking like a, have you ever seen a Kush ball? Do you know what I'm talking about? No? Anybody? Yes? Yeah, it's got like the kind of hard center, and then it's got the little finger things that come out, right? And they all come out because we want lots and lots of branches, because at each endpoint of a branch, you can cleave off a glucose molecule. So very, very quickly, you can have enzyme activity shoot up, you can cut glucose molecules off, put it into the bloodstream. Now we have high blood sugar, and now our muscles can go crazy and we can run a mile and get away from the bear, right? That's glycogen. Okay. So depending on what happens in our bodies, right? If we have epinephrine, we have a surge of epinephrine, what's going to happen is that we're going to stimulate this kinase, this phosphorylase kinase. Well, what does a kinase do? A kinase phosphorylates. It phosphorylates glycogen phosphorylase, which degrades glycogen, right? So you go this way and you degrade glycogen. So we increase our blood sugar and we get away from the bear, right? Okay, so that happens also in not so extreme terms, like when you go to sleep, right? You go for a long time without eating, but your brain prefers blood sugar as an energy source than anything else. And so you will actually start to break up some of the glycogen in your liver while you're sleeping so that you maintain a relatively stable blood sugar. Okay, so what about the other thing, right? We're not trying to run from a bear. We've just eaten lunch. What's your body gonna make? Insulin. Well, insulin is going to actually stimulate protein phosphatase 1. What does a phosphatase do? It removes a phosphate. So it actually removes this phosphate from glycogen phosphorylase. Now glycogen phosphorylase is inactive. So what's going to happen? You're going to inhibit glycogen, GLYs, degradation. And at the same time, which we don't show on this slide, you're actually going to activate glycogen synthesis, glycogen storage, right? So at the same time, you have one up, the other's down. But that's really, really important, glycogen. Okay, so the last one is enzyme activation via proteolysis, right? So zymogens are what we call enzymes that are not active until they are cleaved. So if it is a complete protein, but it's got a piece of it attached to it that is inhibiting its activity, that's a zymogen. When it reaches whatever conditions like low pH that actually induce that cleavage event, then that piece is cut off and now we call it an enzyme. Okay, so a zymogen is the inactive precursor. And then once we cleave it, then it becomes the enzyme. So a lot of times the little piece that needs to be cleaved off actually blocks the binding site. And so that's one of the reasons why the enzyme is no longer active, because it cannot bind, and the substrate cannot bind inside of the active site. And I'll show you a little animation that's really cool with that. All right, so how do you have that? Well, if you look at the entire zymogen, right? So this is chemotrypsinogen because it's a zymogen. Some of these words get crazy, right? You can cleave it, right? And when you cleave it, you're going to make different products that do different things, right? So in this case, you need multiple cleavage events in order to generate active enzymes that will actually break down the food. So I want to show you the animation of this one. I think that's the last. Yeah, that's the last one. I want to show you the animation, if I can find it. How much time we got? 2 minutes. I think it's a 2 minute. 2 minutes and 58 seconds. Oh, okay. But I got no sound. There we go.
In this video, we will explore 2 examples of how specific enzymes are regulated.
Oh, come on.
The bacterial and
Okay, well, there it is.
aspartate transmetabolase, or ATCase, is an example of an enzyme that is regulated with allosteric mechanism. ATCase is responsible for an early step in the production of the nucleotide cytosine triphosphate, or CTP. Specifically, ATCase catalyzes the formation of N-carbamyl L-phosphate from carbamyl phosphate and aspartate. ATCase activity is upregulated by binding to ATP and downregulated by binding to CTP, the end product of the pathway. This regulation ensures that if enough CTP is already present, more will not be made. Note that ATP and CTP bind to a site that is distant from the active site of the enzyme and are thus considered allosteric regulators. When there is a high concentration of ATP relative to CTP in the cell, the regulatory subunits of ATCase bind to ATP. ATP binding stabilizes the active conformation of ATCase, called the R state. The conformation will continually propagate from the regulatory to the catalytic subunits. In the active or R state, the geometry of the active site stimulates catalysis by bringing the carbamyl phosphate and aspartate substrates into close proximity with one another. Once the concentration of CTP has increased in the cell, ATCase will shift towards binding CTP rather than ATP. CTP binding results in shifting the conformation of ATCase toward the inactive T state, effectively shutting down the enzyme. This is an example of feedback inhibition. Protein cleavage, or proteolysis, is another way that some enzymes, particularly proteases involved in digestion and blood clotting, regulate their activity. Pepsin is a digestive enzyme synthesized in the stomach that aids in breaking down food by catalyzing the hydrolysis of peptide bonds. This results in converting long protein chains into smaller peptides. Pepsin is first produced as an inactive precursor or zymogen called pepsinogen. In pepsinogen, the active site of the enzyme is blocked by a 44 amino acid N-terminal protein fragment. The acidic environment of this site causes a conformational change in the pepsinogen that results in it cleaving its own N-terminal segment. The active site is now accessible. These examples illustrate only 2 ways that enzymes are regulated to ensure that they are active at the proper time and place within cells.
All right, and that's it. The end. Y'all have a good rest of your day. Let me end this. Karen, you want to say something?
Yes, ma'am.
Anybody else not get to see their exam? Want to see their exam?
To show you something that I
Yeah.
found. So it's just a video. So gel electrophoresis,
Uh-huh.
small proteins, large ones, because they get stuck. I had a good time.
Yeah.
So I know
Yeah.
for that part.
Yeah. Have anybody? I need to end this. I thought I ended.